Here we have the electron transport chain. In black is our phospholipid bilayer, in red are the 4 transport proteins. Within the mitochondria is the NADH, FADH2, and the matrix. Outside the cell membrane of the mitochondria is the cytoplasm.
The electron transport process begins when NADH and FADH2 each give up 2 electrons. This respectively creates NADH+, a hydrogen ion, and 2 electrons. These 2 high-energy electrons enter the electron transport chain and begin to move through, providing energy for the transport proteins to carry out their function.
As electrons flow through the first transport protein, it then travels along the cell membrane vertically across the other transport proteins. This provides energy for hydrogen ions within the mitochondria to flow outside of the cell membrane. This is an example of active transport, as hydrogen ions flow against its concentration gradient to a much higher concentration of its other ions. As the electron flows through its other proteins, once it reaches its final protein, it is then expelled back inside the mitochondria. Once 2 electrons are introduced back inside the mitochondria, it then attaches itself to the hydrogen ions that are still within the mitochondria. In addition to this, an oxygen molecule that we breathe in also binds itself to result in H2O, which we breathe out.
Now that there is a higher concentration of hydrogen ions outside the cell than there is inside the cell, the hydrogen ions want to diffuse back into the mitochondria. They do this with the help of ATP synthase. And the movement of these hydrogen ions into the mitochondria is an example of chemiosmosis. Now, as the hydrogen ions move through the ATP synthase, their movement causes the rotors to spin and attaches a phosphate group onto ADP, creating an ATP molecule. This is an example of oxidative phosphorylation. When the reaction comes to completion, there are 32 ATP molecules that the cell can use for various energy.